And today I want to talk about one of my projects that involves the identification of genomic regions for protein content in mung beans. There we go. So your first thought might be, why mung beans? So mung beans were actually domesticated in India, and they're considered a pulse crop. And the pulse crops are commonly used for consumption, especially because of their protein quality. And mung beans have a few traits that make them of interest to us specifically, including they have a good flavor profile. They lack this underlying beanie flavor that a lot of pulses have, and that saves processors money because they don't have to process out that beanie flavor. They're very healthy for you. They have a low glycemic index. They're naturally gluten-free. They have a good amino acid profile. They're very highly digestible. They're high in fiber, so anywhere from our youngest to our oldest can eat them. They're also very nutritious. They are high in minerals and vitamins. They also are good in iron content and folic acid. So looking at all these traits, it's no surprise that mung beans have a lot of uses. They're commonly used in Asian cultures for a lot of foods, including they can be used as whole beans in things such as soups. They can be used as sprouts in anywhere from salads to pad thai. They can be ground into a flour and be used in things like milk tea, to produce noodles, or even to make mung bean cakes. And so these are common traditional uses of mung beans, but the US market is also looking at mung beans for use. And one target of focus for them is an alternate protein source. The protein isolate can be extracted and used in multiple products. They use them to make chips. They can be turned into an egg substitute, which is especially of interest with the recent bird flu. They're also used to make healthy snacks. You can actually make mung bean ice cream, and I've actually eaten it. It's actually pretty good. You can also use them in beauty products, which was surprising for me to learn. So the beauty industry is also interested in mung beans and how we can use them. So with all these uses, it's actually no surprise, like global market demands can't currently be met. And so that brings us to the mung bean breeding program here at Iowa State University. Dr. Arti Singh started the breeding program back in 2017, and she saw potential as this crop. It's a short season legume crop, 60 to 90 growing days. And due to that, we can see applications for potential relay, double, and intercropping systems. It's also of interest to conventional and organic markets, so it can be used widely. And farmers from across the United States, along with processors, are interested in this product. We can see that she's made connections with processors all the way out in California to Kentucky, Texas, Nebraska, Minnesota. There's farmers out in California, Kansas, Oklahoma, Tennessee that have contacted Dr. Singh directly asking, like, do you have a mung bean that I can grow in my field? And so this is super exciting to see all this growing interest in mung beans and how they can be used in the US. And so since we're targeting that protein market, my projects revolve around how can we explore this protein content and use this in our breeding program. So first, we wanted to facilitate the development of an NIR calibration equation for high throughput phenotyping. We also wanted to evaluate the distribution of protein content present in our diversity panel. We also wanted to use the wet chemistry-based protein data that we're going to collect to explore genomic regions associated with protein content. And then finally, we wanted to use all of this to provide insights into the breeding program. So first, we had to select, what are we going to use? What's our germplasm going to be? Previously, a postdoc here was able to collect 3,000 accessions from GRIN. We have this amazing resource here in the United States where we can get germplasm. And so we were able to find 3,000 accessions that were planted out here in Iowa. They were tested for their ability to flower and produce seed. And so from those accessions, about 480 were narrowed down. And then a previous postdoc and PhD student did an identity by descent analysis and determined we could maintain that diversity with 375 accessions. So that's what we used moving forward. So those lines were planted out in 3 environments across 2 years with 2 replications in each. And here on the screen, I have an image of our field on the top. So it's one of our GWAS fields. I also have an example of the seed after harvest and then some samples on the NIR, which I'll talk about next. So we harvested these seed samples, and I mentioned facilitating that NIR equation. So we decided, based on previous work, there have been a couple NIR equations developed for protein content in mung beans, but they're typically made off of sample sets of like 1 to 200. They're very low. And if you look at like soybean equations, they're very robust. They are high in sample count. So we wanted to produce an equation that we could have this higher sample count, but we also have to balance that with cost. Getting wet protein data for a large number of samples is expensive. So we decided to go with 5 replications, and after cleaning, that left us with about 1,766 samples. So we took those samples, we scanned whole seeds with our benchtop NIR, and then we ground them into flour and also scanned the flour samples. The motivation behind this is we want to see if we can develop an accurate equation with whole seeds, and if not, then we can grind them into flour, but whole seeds is hopefully the goal. And so from this, we were able to collect a large data set that we can use for training and testing this NIR calibration equation. But we also needed to get that validation data from the wet chemistry. So that flour was shipped out and we got protein quantity via the combustion method. And that's the data I'm going to talk about for the remainder of this presentation. So after we got that data back, we wanted to look what distribution do we have present in our panel. And we were super excited to actually see that we have a pretty good distribution. I was excited to see a normal distribution. And our current check varieties have about a 24% protein content on a dry basis, and processors are looking for a 26% content on a dry basis. So we were super excited to see that we had this wide distribution and a number of lines to select from that are over that 26%. And Dr. Singh was even able to identify some parents that she wanted to integrate into the crossing program, and she's actively using those today. Then moving forward with this data, the previous postdoc that helped put together our panel, he also did genotype by sequencing, so we have this genotype data for these lines. And so I took that data and the protein data, and I was able to run a genome-wide association study. We were able to find 8 unique SNPs, and I used PharmCPU, Blink, and Sven. Sven's a more Bayesian method that's actually developed by a statistician here at Iowa State. And today specifically, I want to focus on this SNP on chromosome 5. And we're interested in it. For one, it has a high minor allele frequency of nearly 0.5. And also we can see this allele difference between our high and low protein lines. So if we look at the high protein lines, that's my top 20% of lines based on protein content. They typically have that GG allele. And then if we look at the low protein lines, those are my bottom 20% of lines based on protein content. And they have a switch where they have that CC allele. And so when we looked into that SNP, we also found that it's within a gene. It's within this Mung bean gene. Sadly, this gene is unannotated. And so I went to BLAST-P to see if I could identify other genes that were associated with it. Maybe they have some information about them. I was able to find that it did align with Azuki bean, lima bean, black-eyed pea, common bean, and even pea, but none of those genes are annotated or previously reported in any literature. I was lucky though, and this soybean gene was reported in previous literature, and I was able to find that first, this gene's expression was increased in nodules when they were under drought stress. And we know nodules are important for nitrogen uptake and protein synthesis. There's also been some studies that look at how the drought pathway and protein synthesis pathway are related. We also found that gene expression, or the expression of this gene, is induced by ABA. And ABA has been associated with regulating the synthesis of seed storage proteins. So this was kind of exciting to see. Soybeans a much more studied crop, as we know. So it's exciting to see that there is some research in a gene that aligns well with our mung bean gene. And so looking back on everything we've done and what we want to do moving forward, we wanted to facilitate the development of that NIR calibration equation for high throughput phenotyping. And we were able to collect a large data set for training and testing. And we're super excited for this. This is what I'm going to continue to work on because we're lucky enough in our program to have a combine that actually has an onboard NIR. So it's super exciting, especially as a breeder, to think you could get yield and protein content at harvest. We also wanted to evaluate the distribution, and we were able to identify parents that could be used in the crossing program, and they're currently being used today. And we also wanted to identify genomic regions, and we were able to do that, and we want to continue looking at potential candidate genes, especially following Dr. Schnabel's talk, and see what we can find, and look to see if we can develop some markers that could be used for potential marker-assisted selection. So these are all providing insights that are currently being used or will be applied in the breeding program in the near future. And I have to thank everyone who contributed to this, especially Dr. Singh, my mentor, and all our current and past lab members, undergraduates, staff members. This was a huge project. Collecting samples, cleaning samples, grinding, scanning, all of it took years to complete. So I have to thank them and, of course, funding agencies. So thank you.
Hi, Ashlyn. Thank you for